Welcome to Common Hour, everyone. My name is Jim Strick. I'm chair of the program in Science, Technology, and Society, and a professor of environmental studies. I'd like to introduce our ASL interpreter, Libby Berlin. Thank you, Libby, for interpreting for us at Common Hour. Please note that if you need to change your screen to include Libby as you watch, you can adjust the screen display mode in the small box labeled View toward the upper right corner of the Zoom window. You can enable the CC closed captioning if you like as well. A very warm welcome to all at our second Common Hour of the semester. Common Hour is a unique and inclusive program that brings the Franklin and Marshall community together every week during the academic year for culturally and intellectually enriching events. It's the only regularly scheduled event that unites students, faculty, and staff, and invites the larger F&M community to join us. Throughout the pandemic, the Common Hour has continued to provide a virtual gathering space and a source of inspiration, education, and compelling discussion for the F&M community and beyond. We hope you'll join us during Common Hour next week for Lydia Slobodian and Lucas Slobodian. Their talk is entitled Swimming with Jellyfish, Building a Startup to Eat More Sustainably. Please follow Common Hour on our webpage, the Presence Events calendar, and our Common Hour Facebook and Instagram pages. During today's event, Zoom viewers can submit questions for our speaker via the Q&A feature. Please indicate your affiliation with the college, but we do not need your name. Now I'd like to introduce Cameron Wesson, Provost of F&M, who will introduce today's speaker.
Thank you, Jim. It's indeed a pleasure today to introduce our Common Hour speaker, Daniel R. Ardia, Professor of Biology and the 2021 recipient of the Bradley R. Dewey Award for Outstanding Scholarship. Dan is an engaging, rigorous, and outstanding educator and scholar whose wide-ranging research program has earned international recognition. His research integrates evolutionary biology, behavior, physiology, and natural history with a particular focus on interactions among stress hormone levels, immune system function, and reproductive effort in birds and fish. Dan's research transcends scale, building out from the local to the continental and global, and has been supported by several major research grants, including a recent grant of more than $2.5 million. Additionally, he's successfully integrated numerous F&M student collaborators in all aspects of his research. More than 80 F&M students have worked with Dan on independent research projects in locations ranging from Lancaster to Wyoming and Alaska. He's provided transformative intellectual experiences for his students and has become a beloved mentor and advisor. As one student wrote, quote, "Working in Dan Ardia's lab was one of the most valuable learning experiences I've had at Franklin and Marshall. I've never felt as if I was simply a research assistant working for Dan, but I felt as if I was working with him," end quote. Dan has produced more than 60 peer-reviewed journal articles and publications that have been cited collectively more than 4,000 times. However, his scholarly impacts extend well beyond his publication record, including his election as a Fellow of the American Ornithological Society in recognition of his exceptional and sustained contributions to ornithology. Dan is also a highly regarded colleague who demonstrates legitimate concern for the well-being of others. That spirit of collegiality even extends to those of us in the administration, including his generous loaning of the beautiful red fox on display in my office after he learned that I had a love of all things fox related. In light of the totality to his contributions to F&M, Dan was recently selected as a Charles A. Dana Distinguished Professor. The title of today's presentation is How Animals Respond to a Changing Environment, a Global and Local Perspective. Please join me in welcoming Professor Daniel R. Ardia.
Did I unmute? Good. Okay. Well, thank you so much for that wonderful introduction and for the honor that I've received today. Very grateful for the support over the years. And while I wanted to acknowledge that I feel I'm deserving of this award, there are many others on campus equally deserving, and I look forward to when they're similarly honored. I also wanted to acknowledge the privilege that has played a role in allowing me to be as productive as I have, and both the explicit system and the implicit norms of science create a system where a white man like myself moves through it frictionlessly, allowing me to achieve funding, publish papers, gather new opportunities, more easily have my voice heard, and to receive and grow from criticism and more. So to get to the talk, we live in a changing world, one where the pace of change exceeds historical rates as we can best reconstruct them, except at times in the past when mass extinctions like we see now remake the geologic and the biological world. My research is focused on understanding how animals interact with this changing world. I'd like to talk to you today about a range of studies, focus on understanding how animals interact with their environment, with the lofty hope that I've contributed in some way to our collective knowledge of environmental change, what lessons we might learn from evolution, and what sense we have for how organisms might respond moving forward. The three themes that underlie how organisms interact with their environment. Biodiversity right now has to deal with rapid, unpredictable change, novel environments that they might never have seen in evolutionary time, and increasingly fragmented landscapes. I'll cover a variety of research approaches to understand environmental change that range from comparing across species, across populations within a species, how organisms respond to novelty, and then finally how species community responds to environmental change right here at home. So the role of the environment in driving environmental change is the key underpinning of evolutionary theory. The interplay between genetic diversity and the diversity of life has given us the wide range of organisms that we see. But it's very hard to tease apart the role of environmental variation when groups do not share in common a common evolutionary history. So for this reason, restricting experiments to a single lineage can allow us to look at divergence from a recent common ancestor to investigate patterns and processes that might help us understand how some species have developed particular kinds of responses to different environmental conditions. I'll outline in brief, and then in a selected experiment, a large-scale study using the genus you see here, Tachycynetta, which is composed of 9 species found across the Western Hemisphere. Right as I arrived at FNM, we received an almost overwhelmingly large award through an NSF program called Partnerships in International Research and Education, in collaboration with my good friend and PhD advisor, David Winkler, based at Cornell. The work allowed us to spend 6 years studying these swallows across Latin America and Canada. We collaborated with a local scientist at each site and eventually settled on 7 focal species. The 4 that are closest to the equator are shown here, the mangrove swallow, Tachycynetta albalinea, breeding in Hillbank, Belize. Tachycynetta albaventer, the white rump swallow, breeding in the Llanos region of Venezuela. Tachycynetta stolzmanni, the Tumbes swallow, breeding range restricted in oak savannas in northwestern Peru. And then finally, Tachycynetta leucoroa, the white winged swallow, breeding near the Rio Parana in Santa Fe, Argentina. Our 3 temperate sites were a focal site for Tachycynetta bicolor, the tree swallow here in Ithaca, New York, the violet green swallow, breeding near Corvallis, Oregon, and then finally down at the far end of South America, the Chilean swallow, Tachycynetta mayani, breeding near Ushuaia. This was an enormous collaborative venture with dozens of field assistants to whom myself and the other PIs owe an enormous debt of gratitude. Many of those assistants were FNM students who traveled the world studying swallows and learning about how to do international research. So the key conceptual underpinning of our work was to understand how environmental variation across latitudes, particularly comparing tropical and temperate regions, drive a range of behavioral, physiological, and ecological traits that all co-vary in interesting and complex ways. So traits that drive how organisms interact with their environment, as I said, can occur in concert. And we can think of them as almost like a suite or syndrome of characteristics. One way to characterize those is along a gradient of what we might call pace of life. So using an extreme example here, we can compare elephants and mice. So elephants would show what we call a slow pace of life, characterized by a long lifespan, high survival, long development periods before beginning reproduction, a low metabolic rate, and a small number of offspring in their lifetime. In contrast, as we probably are all aware, mice live short lives, reflected in their physiology, very high metabolic rates, and significant investment in reproduction as they begin breeding young, have large multiple litters, but each offspring has a lower chance of survival. We can scale down then just to the genus Tachysonetta, which, as I said, best to look within a single lineage that share a common evolutionary history, and look at how latitude as a proxy for environmental variation has helped shape pace of life. We started with the framing that species found closer to the equator were our slow-paced species and had been experiencing, for a variety of reasons, higher survival and less investment per bout of reproduction, whereas birds at the far edges, both north and south, would be considered fast-paced birds that, because of lower survival rates, would be selected to invest a lot in each bout of reproduction. Across the study, we examined a range of traits linked together, looking at first at adult characteristics, so some of the characteristics of adult survival, physiology, oxidative stress. We looked at investment in offspring, and I'll focus on incubation in a little bit more detail in a second. And then finally, a range of developmental trajectories as well. So again, all within this framework of fast versus slow-paced. I'll choose just one example today, and that highlights the work of Becky Windsor of the class of 2011, because of work that she and I did together here in Lancaster on incubation. When she graduated, we hired her to take the lead on studying incubation behavior in a pace of life context across our 7 species. To me, incubation is a great integrator of these interactions between organisms and the environment, because warming eggs against a temperature gradient is highly sensitive, not surprisingly, to environmental conditions, particularly temperature. Doing so is costly for parents, takes energy, and because the optimal developmental conditions for embryos are often higher than those that parents actually maintain, leading to a trade-off. And finally, we know from a suite of other species that the underlying metabolic sensitivity to temperature conditions can vary across species due to pace of life. So a lot is packed in to studying incubation. We kitted out each nest box with a range of temperature measurement methods, which we worked out here in Lancaster. And that was an enduring theme of this project, is that we did a lot of methods development here in Lancaster and then would ship them out to work at these other sites. So we can very precisely measure how much time parents spend in the box and what temperature they maintain the eggs at. So I would say one consequence of managing multiple projects, especially one that generates a large amount of data, is that under the demands of our job here at F&M, I publish more slowly than I might desire. But one consequence, unintended, is that alums can go on and develop new areas of expertise. And so Becky, after graduating, earned a master's. And together, we've developed quantitative methods to characterize our incubation bouts, allowing us to automate analysis of hundreds of data files across these 7 species. We sought, for the purposes of today, I'll frame a simple hypothesis related to pace of life and incubation, namely that the longer-lived tropical species would invest less in incubation than would the temperate species. And in particular, we predicted that the tropical species would spend less total time spent incubating and would maintain lower incubation temperatures. So here are some preliminary data. So along the x-axis are the 7 species, arrayed in absolute latitude, with the tropical species here on the left using warm colors, and then moving to our temperate species shown in colder colors. The proportion of time spent each day on the nest is on the y-axis. For each species, we have a box plot with the mean shown as this black bar. Sample size towards the bottom. And then the letters refer to significant differences between these species coming from a statistical model controlling for a whole suite of other characteristics, including female age, body size, environmental conditions, clutch size, embryonic age, and others. Patterns here are not simple, but for the sake of the original hypothesis I framed, you can see that on average, the tropical species spend less time on their eggs than do the subtropical and temperate species, with the exception here of the violet green swallow, one of our smallest species. It's also interesting to note that the species even with the highest average time, here Tachycena bicolor, about 57%, that those poor little eggs, regardless of who was incubating them, received less than 60% of the day being warmed. As I said, incubation is a trade-off between parents and offspring. There's a lot of interesting biology packed into incubation. Our second prediction, that lower time spent incubating would also be associated with lower egg temperatures, in fact, was not supported. We see the opposite. So all three tropical species shown here maintain higher average incubation temperatures than do the subtropical and the temperate species. As parents can carefully modulate incubation temperatures, work I found working with tree swallows over the years, these results cannot be explained simply by warmer temperatures in the tropics. We've been following up on this work to try to link these developmental temperatures to thermal sensitivity of embryos with some field methods for this project and its impact on nestling development and physiology. So for this section of the talk, I used a pace of life framework to understand the impact of environmental variation along a latitudinal gradient, using latitudinal natural environmental variation as a proxy to try to understand how different environmental conditions shape the characteristics that species show. Those were intense years at the start of my time at FNM, trying to juggle the diverse responsibilities of my job here, actively conducting local field work in Lancaster on tree swallows while trying to coordinate across all those field sites in Latin America. There are a lot of Skype calls, frantic sending of equipment, constantly tweaking protocols for the field and the lab, and getting to travel to Latin America for field work as well. But having the opportunity to work locally at the same time was really exciting, to be able to follow up on results we were getting from the field and then develop methods and techniques we could send out. I thank a very large group of students who worked here in Lancaster with me studying swallows. We sought, as I said, to dive deeper into understanding the trade-offs between parental incubation conditions and embryonic development. I don't work with swallows locally in Lancaster anymore because of this animal right here. All of our nest boxes were overrun by snakes over the years. And while this is a king snake, it was actually black rat snakes that destroyed the population. But we did find some interesting things, and I'm going to tell you about some of those right now. So I want to go into a little bit more detail here about how early life experiences during incubation may or may not program later life behavior or physiology. So here's an image here of some hatching tree swallows, some still in the egg, some just out, naked, helpless, begging, with some of our temperature monitoring material in there. What kind of information did these little nestlings receive when they were eggs about their environment, and how does that affect their later life? So the overall hypothesis was, how does early life experience program physiological and behavioral performance later in life? And so I wanted to find out whether this environmental temperature information that embryos would receive could either program them for later life acclimation to what future conditions might be like, or if they're pushed too far, could it be a cause of suboptimal performance? So the basic design of this experiment, conducted over a couple years, was to start with two treatments during embryonic development, during incubation, where nests would either be cooled by using this thermoelectric device you see here, if I can find my cursor. So we have one group of eggs that gets chilled during incubation and another that experiences control conditions. Then after the chicks hatch, we cross-foster some of them among nests in order to break the relationship between what parents experience and what the eggs experienced. And then they undergo a thermal challenge later to measure their thermoregulatory ability. It's important to note that when they were nestlings, they were not being cooled. So the effects we would see in nestlings would be driven purely by what conditions they experienced when they were incubated. So then when they were chicks, they were given a thermal challenge in the field. So they were put into this gray cooler in the back of my minivan while I recorded the change in temperature and their metabolic rate through this flow-through system running off a generator. So here again are our little chicks. So the first prediction would be that when chicks are up 7 days old, which is about intermediate age for tree swallows, that they're just beginning to ramp up their machinery in order to maintain body temperature. They're not great at it. It's very driven by developmental stage. So the first prediction would be that this embryonic cooling would reduce the physiological development of younger nestlings so that these cooled eggs would be behind as chicks. But as they developed adult-like thermoregulatory abilities, that these chicks would have an advantage, that they would have been programmed, so to speak, by this early life pulse of cold in order to develop adult-like capabilities earlier. In other words, it would cost them less energy to develop their thermoregulatory ability. I should mention that this idea is not one that I came up with, although it's relatively new among organismal biologists, because the poultry literature or poultry scientists have been doing this for years in order to acclimate chickens to cold temperatures if they're raised in cold areas or warm temperatures. So if you pulse that information in during development, when they hatch, they develop either cold or warm adapted phenotypes. The goal was to find out whether that was occurring in a free living bird like the tree swallow. Okay, so here are the data for the 7-day-old nestlings, which, as I said, are just developing their abilities. So we have the treatment they experienced during incubation. They were either cooled or experienced cold or control conditions. And then on the y-axis here, we look at the rate at which they lost their body temperature because they're not able to maintain body temperature against a thermal gradient. They slowly cool. The nestlings that experienced cooled treatments actually lost their body heat at a higher rate, suggesting that they were behind in development. So this shows that the first prediction that embryonic cooling would reduce physiological development was supported. The key prediction was whether they would have higher performance as they got older. And so now here we have measurements from 12-day-old chicks when they've just begun their ability to thermoregulate. Again, whether they were cooled as eggs or whether they were control conditions. And then instead of looking at cooling rate, what we're looking at here is how much energy it took to maintain body temperature. So if performance was roughly similar, then how much energy does it take for them to maintain that temperature? If they're better at doing so, you would predict that the energy required would be lower. And indeed, that's what we find. So increased energy to maintain that temperature have a higher average value, statistically different for nestlings that experience control conditions than those that were cooled. So this seems to support the idea that in this case, environmental temperature is a potential programmer that can help guide future development. But of course, this information is only useful in an evolutionary context if the cue, early life conditions, in this case cold, are actually correlated with later life conditions. We know that as climate has warmed and become more variable, that this link between the cue and later life conditions have been broken. And of course, we know that under extreme weather, the intensity of embryonic cold or heat could shift from being acclimatory to simply being negative, leading to work that to follow up on that'll come back full circle towards the end of the talk. So to give a bit of a chronological arc to the talk at this point, I had two parallel tracks at play in my grant writing. First, work focused on following up on the results I just gave you about this early life programming, while also trying to continue support for the geographic variation work to be able to work on a broader environmental scale. Because of the constraints of a school like ours, working across a wide swath of geography really does require extensive collaboration. And so at this point, the next step on my research trajectory would be determined by which grant proposal was funded first, a collaborative geographic variation study or a local intensive temperature manipulation study. The geographic variation piece came through first, and we received an award to compare across multiple populations of tree swallows. My collaborator on this work is my good friend, Maren Vitusik, now at Cornell University, interestingly in the same department in which I received my PhD, working at the same site in which I worked as a graduate student. And so one challenge that this pace of life work that I gave you earlier shows is as so much covariation across traits, so much environmental variation across sites, and working with multiple species confounds evolutionary history. Maren is a very focused collaborator, and she convinced me that we should focus on a single key indicator, stress hormones, and a single species, Tachysonetta bicolor. And every time I would bring up, maybe we should measure this, she would say, we should not. And so I'm glad she was right. We kept ourselves focused. Okay, so first let's talk about stress. So in the short term, stress works to marshal resources and help focus us during an acutely challenging moment. Secretion of stress hormones triggers a cascade of physiological and behavioral responses that serve, to oversimplify things a bit, to prioritize our survival over other activities. As we all probably know, chronic elevated stress can be debilitating. And so for free living organisms, it's critical to invest in reproduction, even under challenging conditions, as individuals that fail to breed will, of course, leave no offspring to the next generation. Of course, one would expect that the trajectory of our stress responses would be subject to trade-offs with a whole range of factors, including environmental conditions. So we can look at the interaction between environmental variation and environmental differences and stress trajectories, which might be directly stressful or could drive changes in other factors, such as resource levels and predation risk. So the classic trajectory of a stress response in a bird would look something like this. Individuals would have a baseline level of circulating stress hormone that might vary seasonally or across time or among individuals that represents just what it sounds like, a baseline. Under an acute stressful moment, such as the sight of a predator, hormone levels would increase, would trigger a short-term response that would allow an individual to deal with a crisis. But those elevated levels cannot persist, so consequently negative feedback processes can occur that would serve to reduce those elevated levels back towards a baseline. A lot of interesting biology can be packed into these three metrics to understand individual and population level variation. How might baselines differ across individuals and across sites, the standing defense, so to speak? How does an acute threat raise stress levels? In other words, how much capacity is there for dealing with a crisis and how does that vary? And finally, is there variation in the rate or the magnitude of the negative feedback with this recovery phase? We settled on 4 locations, all within the species tree swallow, Texneta bicolor, the one that we have around here in Lancaster as well. We chose 2 sites that are relatively benign, Ithaca and Chattanooga, Tennessee, where conditions were relatively warm and the breeding season comparatively longer than the other 2 sites. And we chose 2 sites that were more challenging, a high altitude site on the Bighorn Plateau in Wyoming and the Wrangell Mountain Range in inland Alaska. And in both Wyoming and Alaska, the ambient conditions were cold, storms were frequent, and the length of the breeding season was short, leading to strong selection pressure to invest heavily in a bout of reproduction because of lower survival rates of the birds. As I mentioned earlier, conducting this kind of fieldwork involves a large team of people, and we certainly had that. Lots of dedicated field assistants on this project, including a number of F&M students. And one glorious aspect to this project was Cedric Zimmer, shown here, the postdoctoral fellow who moved from France to lead the fieldwork. It was a joy to get to know him, and particularly fascinating to watch his insights into the United States as he worked from a gradient of Americana, survivalist Alaska, to the rural Rocky Mountains, to East Tennessee, and finally to hippie Ithaca. I give enormous gratitude to Cedric. In the project, we tested a range of predictions related to negative feedback, both within and across years, to test whether it was repeatable across individuals, whether it was heritable to offspring, whether it showed correlations with a variety of other behavioral, physiological, and fitness effects in the face of both short-term and also long-term chronic challenges. But for the purposes of today, I'd like to focus just on our most fundamental prediction, which was site level variation. So our prediction was first that Alaska and Wyoming, as the more challenging locations, would have higher baseline levels of stress, that living in these challenging conditions would lead those birds to have higher induced response, so they would elevate their their acute moment in order to respond to a crisis, but that they would be unable to maintain those levels, and consequently they would have much stronger negative feedback to allow them to be able to allocate resources towards reproduction, even when survival might be important, knowing that those sites required birds because of those lower return rates to invest more in reproduction. Okay, so what did we find? So we measured stress levels in birds at 3 time points, in the middle and at the end of incubation, and then caught females again during the nestling provisioning period. So each of these represents an average value of breeding female birds caught at each of these sites. The colors and symbols relate to the 4 different populations. And just as before, the letters refer to significant differences between means when controlling for lots of other variables in a statistical model. Results are not simple, but they do conform at least roughly with our predictions. So if we look first just at the baseline levels, so these would be the levels of circulating stress hormone, shown on the Y, that we would get in the birds before they responded to a stressful moment. You can see that on average, Wyoming and Alaska birds maintain higher baseline levels than do the birds in Tennessee. When they're given a short-term stress, catching them or exposing them to a predator, all four populations elevate their stress levels, but with much higher levels in Wyoming and Alaska, and particularly in this high altitude site in Wyoming. Then if we can look at the negative feedback phase, where we give the birds exposure to dexamethasone in order to stimulate negative feedback, we find that all four populations show a decrease, but a much stronger magnitude of decrease between Wyoming and Alaska, conforming at least more basically with our predictions. Good news is that these phenotypic results in the hormone levels were supported by differences in gene expression in the brain across sites that corresponded to receptor levels, suggesting a link between the physiology and the genetics of the birds. So taken as a whole, our results here show that differences in environmental conditions can drive underlying population differences in a fundamental aspect of physiology and behavior, circulating stress levels. This suggests that environmental conditions, as they become more difficult and more unpredictable, that the underlying genetic variation in a population will be critical. As a subset, hopefully, will be able to shift their responses to meet this trade-off between balancing short-term stress that allows enhanced survival versus the cost of chronic suppression of reproduction. To be honest, the results from this study have been hard to put into a simple context. Working at 4 sites introduced so many variables that it's been difficult to piece out just exactly what the underlying mechanisms are. And so even though complicated fieldwork has a compelling draw for both me and for Maren, who both of us thrive on novelty and travel, we felt that it was important for the next step to retrench to a single population. And so we decided to focus our next grant, which we just received, on work just in Ithaca, New York, with the goal of integrating the insights from the stress work from this study I just described with my earlier findings on developmental temperature. So we know a lot about tree swallow breeding in Ithaca, and one trend that we see is that birds, many species of birds, but especially tree swallows, are advancing their timing of breeding as temperatures warm. So here we have, each dot here represents the first and the median lay date for a particular year of the populations of birds breeding in Ithaca, arrayed up to 2015. And you can see, if we look regardless at median or first lay date, that on average the birds breed earlier. There's quite a bit of variation, which is predicted by spring temperatures, but that the birds have advanced their day of either the first egg or the median egg for the population by as many as 10 days. So the birds are breeding much earlier. They're using a cue, temperature, that in the past has been linked with a fitness outcome, food availability during the period of peak nestling demand. And it's important to restate that. So it's not how much food is available at the time that they begin laying eggs, but rather a month or more later is when the peak food needs are. And so a cue, temperature, has been linked over evolutionary time with food availability. Could that cue break? In other words, does that cue fail to be a useful predictor of when the best time to breed is? And that's what we want to test, whether that's happened and what the consequences are. So here are additional data from Ithaca. And so we're looking here at another way to represent the data. This is just a frequency distribution now of when nestlings are at their peak demand. The dark bar here represents the average value for those. And so if we look at the period from 1972 up to a more recent 2015, you can see that that period of peak nestling demand has shifted from around the middle of June to the beginning of June. And then what's plotted here is the probability of a cold snap. So a cold snap is a period of 2 to 3 days where the weather is too cold for insect activity and thus too cold for swallow food. No swallow food for parents, and especially no swallow food for nestlings. So if we just look at the intersection of when this shift in the timing of breeding has occurred and how it matches up with the probability of a cold snap, you can see that the chances of a cold snap have increased considerably. And so our goal here is to look at how these changes in temperature, particularly the risk associated with increased environmental unpredictability, drives changes in how parents respond and how nestlings respond. So we're going to use these same thermoelectric devices I mentioned earlier to cool temperatures and look at parental feeding and how that affects nestling physiology, both in a single year and over multiple years. So wish us luck. So I want to revisit the idea that there's sort of three main changes that organisms experience in our modern world. One is that the rate of change of the environment has increased, but the other two is that the world has become increasingly novel, that organisms are encountering conditions that they've never experienced before in evolutionary time. And the third, which I'll get to at the end, is that our world is becoming increasingly fragmented. So I've given you what is an otherwise logical trajectory across my research projects of studying environmental variation by looking at geographic variation and by directly manipulating temperature. But sometimes science works in serendipitous ways. And so over years of collaboration with my longtime friend, Marty Martin, shown here, the University of South Florida, navigating local hazards in rural Kenya, he cooked up a plan for me to join his ongoing research on the physiology of invasion. So Marty had been using the course of invasion of the house sparrow, shown here, a bird native to Europe, but introduced into East Africa, as a case study for understanding how species adjust and adapt to novel landscapes, in particular the landscape of disease. We know that house sparrows first invaded Kenya along the coast, showing up here in Mombasa decades ago. And then thanks to attentive naturalists, the timing of house sparrow movement across the country is well known. Marty and his students have been finding interesting patterns that individuals at the range edge, in other words, those individuals that were either actively dispersing or those that settled there and survived to reproduce, appeared to encounter new pathogens and respond more strongly to them. So I joined Marty to visit these three sites that we focused here to see whether or not the cost of mounting an immune response and the amount of energy that the birds invested in dealing with novel pathogens would differ across their range. My aspect of the project was to study the metabolic rate of a fever response, and I'm calling it a fever response, but it's actually a whole suite of physiological and immunological changes that organisms undergo in response to a general immune threat. So we stimulate that in the birds through an injection. So at each site, we would misnet birds, rent a hotel room, capture them and put them in cages in the bathroom, as Marty's grad student Holly Kilvitis shows us here. And then my job was to stay up all night with the birds, moving them from chamber to chamber and measuring how much energy they invested in fighting off a health challenge. Okay, here's a picture of us processing birds in the field. And here are our data for the three sites, ranging as a distance from Mombasa, away from the coast, the distance correlating with the time since invasion. We have three time periods. We have before we gave the birds an injection of lipopolysaccharide, the protein coat of a bacterium that stimulates this generalized immune response, including a fever. We give them the injection and then we measure their response 24 and 48 hours later. And then on the y-axis here, we have the metabolic rate that they show in response to that elevated body temperature. First thing to look at before the injection is that there's differences across sites, with the sites farther from the coast, the birds maintaining a higher resting metabolic rate than at the coast. This is consistent with knowledge from other invasions where the subset of individuals that are found at range boundaries are those that are most active, most likely to disperse, which is likely correlated with metabolic rate. Second thing you can see is that when you give the birds an injection, they raise their metabolic rate, raise their body temperature, suggesting that the overall design was sound, and that we have a much higher, in fact, the highest level of fever response on behalf of the birds is the site farthest from the point of colonization, suggesting that the subset of individuals at that site are the ones that are investing the most in an immune response when facing novel pathogens. And interestingly, out at Nakuru, the site farthest from the invasion spot, still maintain elevated metabolic rate even 48 hours after the injection, while birds in Mombasa recovered. As this was just for a single site, we sought to test the generality of this pattern by visiting Senegal and West Africa, where birds also invaded and were found along the coast here in Dakar. And again, attentive naturalists can tell us what the course of the invasion, and they moved up the coast and then up the Senegal River. We visited 3 sites to gather data for a grant proposal. We'd also planned to add additional pilot data from Angola in 2020, but those plans have been on hold. Here's a picture of one of our field sites in Senegal, just in case you thought we were in exotic locations. House sparrows are human-focused birds. They can't thrive in natural environments. Rather, we catch them in people's backyards and abandoned lots, which leads to many fascinating conversations with bemused and interested passerby. Why would you come all the way here to catch these trash birds? As a final image, here's one of our field labs where we processed blood samples. Taken as a whole, this work on the invasion physiology of house sparrows gives us some insight into that as organisms face novel environments, that a subset of individuals who have the genetic variation required to match this new and changing environment will be the ones that persist. And in those species that do not have that genetic variation in the face of a novel condition will be those that are unable to shift their range or will go extinct. The last piece I'd like to talk about today is shifting to work more locally. And as I said, interested in rapid change, novel environments, and then finally the world has become more heterogeneous, more fragmented. So this is a map of Lancaster County, and it shows the highly fragmented nature of our landscape. You can see very few blocks of forest, and those remnant patches that are hanging out here and here are all surrounded by a potentially inhospitable matrix of agricultural land and urban and suburban sprawl. In fact, Lancaster is among the least forested counties in the state. I had come of age as a graduate student when fragmentation research was one of the most important pieces of conservation biology. I'd always hoped to be able to set up a long-term study to study fragmentation right here in Lancaster as a way to give students research opportunities through the full cycle of field biology and designing the experiment, planning out field logistics, doing the field work itself, managing large amounts of data, and learning to run complex statistical models. Initially, as a trained ornithologist, I'd hoped to do that work on the movement of wintering birds, and many students worked with me for many years capturing wintering chickadees up at Millport Conservancy, and I thank them for all the time we spent standing around, keeping ourselves warm, checking the nets, and waiting for the discovery of a bird in the net. But that project proved too difficult to sustain, so I shifted gears to focus on the forest mammal community here in Lancaster and now York County, in collaboration with Lancaster Conservancy. Enormous gratitude to them for letting me work at their sites, and in collaboration with Eric Lonsdorf of the Natural Capital Project. So since 2016, my students and I have collected over 660 camera placements all across Lancaster and York counties. And it has been all I could hope as the students involved have really had an opportunity to see a field biology project right from planning all the way to data collection, data management, and hopefully to collect long-term data useful to local conservation. I'm grateful for all their hard work, for being able to find time to go to the field, even when they're busy with their other classes, and getting a chance to experience the beautiful nature right here in Lancaster. So the processing of these images for analysis, we're up to about 2 terabytes of photos, which is about 1 million plus images, has proven to be the last challenge to producing a full analysis. But with the help of Corinne Richter, class of 2022, we're close to a machine learning solution, and by we, I mean Corinne, to detect and classify images. And I wanted to give a shout out to Jason Brooks and Tony Weaver for their assistance in helping us move forward. This part of the project has really been a game changer. I'm going to show you some preliminary data and of course, lots of pictures. But first, here's a pretty picture of our landscape reflecting the various land uses, with more urban areas shown here in red around Lancaster City. And so the core research question I was interested in was trying to study the presence and abundance of mammals on 3 different scales. The area close to a camera, which could be influenced by things like what forest type, the topography, distance to a stream, the area of a patch or the shape of a patch, and then what surrounded it in the matrix. So sort of 3 different scales of consideration. So here are just some images of some preserves that you may have visited here in Lancaster. So here's Tuckwan Glen, and then Reed Run, now part of Clay Clark Nature Reserve. It may look to you like a relatively big block of forest, but if you consider the shape of it, you can see that there's lots of pieces that push into this habitat and that it's surrounded by a potentially inhospitable matrix. The only large block of forest left in Lancaster County, other than at the border of Lancaster and Lebanon counties, is Welsh Mountain. It's the only spot left in the county that's considered core habitat, large enough to hold a subset of original species, but it's still surrounded by a matrix of non-forest. And many patches are indeed just patches, like these preserves here, Shiprock and Holly Point, that are islands surrounded by a sea of agriculture and suburbia. I was initially motivated to characterize the incredible versatility of the red fox, which I see close to campus. How did these cool and adaptable creatures survive in an urban landscape? We'd hope to be able to observe the impact of coyote expansion on red fox numbers, but we pick up coyotes infrequently for a variety of reasons. We've tried hare snares with some moderate success, but coyotes, as Sarah Dawson can tell you, are very hard to work with. Here, just for your interest, is a coyote marking its raised woods. All right, so let's look at some results for the first 3 years. So this comes from about 300 camera placements. Each point on the graph represents a location in which we placed a camera. So because our landscape is highly spatially correlated, which really just means that some land uses co-occur, like urbanization, both heavy and light urbanization, or that housing developments tend to be found near open space, the measures need to be combined into an index that corrects for that covariation. So along the x-axis, we see a degree of forest cover with camera placements here at locations that have more human development, and here locations here that have more forest. And then on the y-axis is an index of biodiversity. That's a mix of both the number of species that we find and how they're distributed. You can see that there's a lot of variation at any degree of forest cover. There is a wide variation in biodiversity. And while we can fit statistically align to this, we find very little impact of development on biodiversity. Does that mean that animals don't care about human development? No, not at all. What it means is that any forest sensitive species that used to be found in Lancaster County, like a bobcat, a fisher, large populations of black bear, even gray fox, which is relatively rare, they're mostly gone. Porcupines, gone. And so what we have left are just the survivors that have already learned to persist or thrive in this rapidly changing, novel, and highly fragmented landscape. So the general effect we see are differences in abundance, but not differences in diversity. So given this human domination of the landscape, the project has really morphed to be more about people interacting with environment or animals. And so we can ask whether certain kinds of human activity have impacts on animal behavior. And so Lancaster Conservancy, many of you may not know, started as a group of interested hunters worried about the loss of forest. So a significant number of Lancaster Conservancy preserves allow hunting. So we could use our cameras to estimate hunting pressure and look at the behavior between preserves that allowed and didn't allow hunting. So here we see seasonal changes along the x-axis, and these data come from a period when the cameras were spending the summer in other places. So there's data from the spring and fall and winter, but not from our summers. Plotted with the proportion of activity of total sightings of deer that occur either at night or during the day. Broken down between preserves that allow hunting and those that don't for the months in which hunting is allowed. So if we look first just at the period in the late winter and the spring when no hunting is allowed in Pennsylvania, the deer on average are actually quite active during the day, that they spend no more than 20 to 30% of their time active at night. When the fall comes at both sites, activity at night increases, but that at those sites that allow hunting, particularly in the busiest hunting months of October and November, the deer shift to be much, much more active at night, suggesting either that the animals are making an adjustment or that those deer that fail to be night active are the ones that get killed. So one hard part about doing work in human-dominated landscapes is this work is just correlational. We can't allocate sites to treatment. I can't ask Lancaster Conservancy to let one site be hunting allowed for 5 years and then do a swap. But it does suggest that deer are responding very carefully to hunting pressure. Another trend we see is we see a lot of cats. More cats than most would make me comfortable, and they don't often quite far from your house. So this is my public service announcement to keep your damn house cat indoors. If you care about birds, don't trick yourself. Your fat, slow house cat still kills birds. Keep them inside. They travel long distances into the forest. We pick them up in the camera, literally hundreds and hundreds of meters from a house. So keep your cat inside. Thank you for listening. Okay. So as I mentioned, the project really is about people and animals. And with the pandemic, people have rushed to the woods. As you might know, Tuckahoe Glen's parking area has been closed. It's been redone. People just simply could not abide by having a closed parking lot, would leave their car wherever they wanted. And we see people in the woods at crazy rates. I can tell you anecdotally from looking through thousands of images that preserves where we might have seen 3 to 5 parties on a weekday have shifted to 3 to 5 parties per hour. And on weekends, the most popular preserves like Kelly's Run might have 8 to 10 groups per hour. So there's people on the trail from dawn to dusk. What do wildlife do? Well, they appear to shift to be active only at night. You can look through cameras at places like Kelly's when they're busy and you never see an animal. The other effect we'd like to test is the effect of dogs, whether dogs are there and whether they're leashed or not, can understand human activity. So if you see one of our cameras out there, please do wave hello. So to finish up a bit, all of this work working locally has gotten me much more interested in the ecology of Lancaster City and of the Conestoga River. And in fact, now I teach a course in urban ecology, a great example of how our scholarship works hand in hand with our teaching. Many of you may have participated or offered to participate in our studies looking at urban bats in the landscape, and Juliana Stoll is working this year to test the effect of green roofs on buildings in Lancaster City as a way to support urban bat populations. Also interested in working more closely in Lancaster City to understand socioeconomic disparities in access to green space. Yes, Ella, this is your map. Extensive evidence shows systemic inequities in access to green space across urban areas. And we've been trying to understand whether that occurs here in Lancaster. So this map shows the census tracts in Lancaster City, colored by socioeconomic status, with green being those tracts characterized as economically vulnerable by a recent analysis by F&M's Floyd Institute. And the red areas on this map here are those that were redlined in Lancaster in the past, a key predictor in other cities of socioeconomic impact on green space and environmental quality. So Ella Murray, as I mentioned, class of 22, is working this year to develop maps integrating green space characteristics, level of protection, whether they be formal or informal spaces, spaces without protection, and socioeconomic status, hopefully to contribute to Lancaster's comprehensive planning process to think very carefully about who has access to green space in Lancaster. So for this last part of the talk, my goal was to integrate research across these multiple spatial scales to better understand how people interact with their local environment and the effect of wildlife to inform conservation and management of my home right here in Lancaster. I've been very fortunate to have an amazing family which shares my passion for the outdoors, although less now as they're teenagers, and indulge me in my quirks. My parents have given me the greatest gift I could ever have hoped for. It's unconditional support and belief in me through my ups and downs. And special gratitude to Ellie for joining me on this journey. I love you. Thank you to you all for listening, and I'm happy to answer any questions that you might have.
So, audience, remember again, you're welcome to submit questions through the Q&A function. And I do see plenty coming in already. The first question comes from somebody identifying themselves as an administrator at F&M. Fascinating information, thank you. There are several feral cats in my area. Many of my neighbors feed these cats, which also hurts the nature preserve in our area. Do you have any suggestions on how to address this with my neighbors? I'm new in town and would like to do this discreetly or anonymously.
Oh my. I mean, I think that most people who love cats don't necessarily know the impact on birds, but the most important thing that you can help people who feed feral cats to understand is that the life of a feral cat, even if you provide supplemental food, is short and brutish, and that the most humane thing to do for those cats is to either rehome them or move them indoors or even euthanize them. That you're not doing the cats any favor to leave them out there struggling with the elements.
I hope that makes sense to your neighbor. Next question. You mentioned that predation on eggs and hatchlings by snakes in your Lancaster field sites led you to stop doing incubation research here. Was there an increase in local snake population sizes? Were snakes learning to visit the nest boxes, or was some other factor driving the increase in predation that you saw?
That's a great question. It's more the second that snakes found our boxes. So most of the boxes were at Lancaster County Central Park, where they were reasonably close to the woods. And so what we found is that the first year, occasionally we'd see a snake, and the second year we'd see more. And then eventually it would be, you could literally watch the snakes trap line, and they would go from box to box, crawl up, look in, eat whatever was there. If not, drop down and keep moving. Predator guards, which work reasonably well, it's very hard to keep black rat snakes out. So a 6-foot plus rat snake could hold its body on the pole, go around the guard and go in. I personally found it quite fascinating, but it was pretty hard on the students, when we'd have nests that we'd worked on for weeks, and then you would open the box and there would be a snake wrapped up in there, and you could literally feel on its belly, 5 eggs or even 5 nestlings, that were all just right in there. So it's, part of nature. But
I feel like I'm reliving the squirrels, figuring out everything I do to try to protect my bird feeders. We really do underrate the intelligence of animals in the wild, don't we?
we do. And squirrels, even though I don't care for them much, I have enormous respect for their problem solving. So
everybody has to go to the YouTube video of a retired NASA engineer who sets up an American Ninja Warrior course for squirrels that they have to go through to get to his feeder.
So actually, I want to give a shout out to the Common Hour archive, back to the cat question. So years ago, my good friend Pete Mara, who's written a book called Cat Wars, came and gave an in-person Common Hour that's available in the archive. And I would encourage you, to answer your question, to watch that. And Pete gives a lot of good information about cats and birds. So
I remember it was extraordinary hearing the numbers of birds that are eaten. When you work collaboratively with international partners, how does an episode like the COVID pandemic impact your work? Does using Zoom minimize the impacts, or do you have sites that just shut down entirely?
that's a good question. I mean, the Latin American project had been over many years pre-COVID, but I know for colleagues that have worked internationally, it's been a challenge for getting to field sites, a challenge for collaborators being able to move around, for getting equipment, for permits, for getting through things through customs, for graduate students, from the collaborative country to come to the United States for any reason. So it's really, especially in the first summer, it really shut things down. But I think that most schools, including our own, have realized that we can't just, close research down completely, that we have to find ways to balance people that do field work, whether it's in the sciences or other disciplines, that they need to be able to get out and do their work and work with collaborators and go forward. So
this question comes from a faculty member. Describe the process of getting a sample from a bird in the field and then turning that into a numerical measure of stress hormone levels. Is it a blood sample? Do you have to freeze them to get them back to a lab? Do you measure the hormone levels yourself, or do you have chemistry collaborators who do it, et cetera?
yeah, great question. So it is a blood sample. Sorry to not say that. So the birds, we catch them and we take a small amount of blood. The getting of that sample within a short period of time is really critical in order to feel like you've gotten a baseline. So there's a lot of stress on the part of the researchers in order to get those samples quickly. Depending on where the work is done, the samples, we cool them off as quickly as we can. So we bring ice to the field. If we have access to electricity, we'll rush them, put them in a cooler. The work we were doing in Africa, our blood samples, we actually would have to carry a liquid nitrogen dewar in order to get the samples cold really quickly. Then when you bring them back to the lab, if they're frozen, they can stay for a while, though it's not ideal. And one can process the samples pretty straightforwardly. We've done some of that here. So shout out to Meredith Bayshaw for letting us use her hormone lab over the years. But at this point, we do it all at our lab. We have equipment at Cornell where Cedric was doing the samples.
So overnight post, I guess.
Yes, or one freezes them and then packs them in their suitcase and hopes they stay cold when you fly them back.
What are some frustrating obstacles that you've had to overcome while working with wild animals in the field?
I mean, the weather is by far the most important. As I mean, it's ironic that we're studying environmental change, but we want the environment to be within a particular set of boundaries. And when you've got a big experiment going and one of those cold snaps comes in and just wipes the entire population out, if the experiment required 3 weeks and they're 2.5 weeks in, and then you lose all your birds, that can be pretty demoralizing. As a graduate student, that was much more stressful because the short-term stakes were higher. I feel like a place like F&M, it's a downer, but it's, on the other hand, it's just part of doing field work. So yeah, the weather is by far the hardest. So
you're making me realize that psych grad students need to have stressed field biology grad students as sources of hormone samples for baseline stress levels. I think we have time for one last question. Do you recommend snake population control? We have lots of rat snakes. We fear our efforts to attract birds might be leading them to their deaths.
I mean, I love snakes, so I wouldn't advocate that. And certainly, snakes are natural populations. I think that, carefully siting nest boxes in retrospect, we could have found more open fields. We have nest boxes that Andy DeBet built that are out at Baker Campus, Spalding Conservancy, and we don't, as of now, see much snake activity there. So maybe we made good choices. So I think it's, it's a balance.
Well, I don't know how many other snake lovers there are in the audience. I hope some. I'm joining you in that. We're at 12:30, and I think we should thank our presenter. I'm mindful of students who have class and professors who have to go teach them. So many thanks to Professor Ardia. It's really a fascinating talk and a really interesting insight into what it's like to be an active researcher, especially at a place like Franklin and Marshall. Thanks again to Libby for interpreting. I want to just remind you of our Common Hour talk next week. Swimming with jellyfish, building a startup for eating more sustainably. I think environmental types will definitely be interested in that one as well. But anybody who wants to look into more sustainable startup companies probably also find it interesting. I hope to see you all next week, and thanks very much to the audience for your participation. And let's keep Common Hour going.